Hey everybody, chapter 15 is going to be dealing with specific immunity. So that's mostly going to be like the B cell, T cell stuff and how like your body is going to respond to specific things. We already know that in innate immunity, in the non-specific immunity that we talked about in chapter 14, that your body can recognize general patterns as being bad. Those PAMPs, remember, I can never remember what it stands for. So pathogen-associated molecular patterns. Yeah. Anyways, it can recognize stuff like that, or it can, there's other things as well that will trigger it to start with inflammation and all that stuff. We talk about the complement cascade and everything. But now we're going to get into how to control that very specifically, because you don't always want to just mount general attacks, right? So sometimes you want to actually be a little bit more specific. So especially long-term. So we're going to talk about how the immune system is able to be diverse, and we're going to outline the steps involved in the development of the immune system itself, differentiate between the results of cell-mediated and humoral immunities, and then explain the principle of vaccination and how it works in our benefit. So we're getting back into this far right side. Getting into this far right side over here. Oh, I need to do that. Don't let me forget. With the acquired specific immunity stuff over here. So we're going to be getting into all of this. And I know that you're like, okay, well, that looks like less, but it's not. Believe me. All right, specific immunity. We're talking about the first step being lymphocyte development, presentation of antigens, and the antigens are going to activate, challenging the B cells, challenging the T cells, and that's going to activate them to either produce antibodies for the B cells or lead to cell-mediated immunity with the T cells. So we'll get into what that is. Don't worry. This first step in all of that was the lymphocyte development. Lymphocytes are developed from precursors in the bone marrow. We talked about that last time. That leads to a certain lymphocyte, like stem cell precursor that's specific to lymphocytes. Then we have special bone marrow sites within your bone marrow for like B cell development, whereas T cells are going to mature and develop in the thymus. Either way, they're both going to end up going out to the lymph nodes and the spleen, essentially, in order to interact with pathogens or antigen-presenting cells and stuff like that. So first we have contact with an antigen. That's what this blue guy is, the blue blob. Our cell recognizes that antigen. Essentially, it interacts with that receptor on the surface. Okay, they're called MHC markers. We'll get what that is in a moment. And then it's going to display that to a T cell. See, there's a T cell down here. Okay. So T cells are going to come along. They're going to see this, interact at this junction here. They have another receptor to interact with that T cell receptor. And then that's going to activate the T cell to do other things. Well, what is a T cell? This is T helper cell. This T cell is going to then stimulate B cells. What's important about that? Well, you'll see. Essentially, we'll stick with the B cells for now. The B cells are going to be activated now. They can differentiate. They can turn into either plasma cells that secrete antibodies. That's the whole point of most of this, right? And then memory cells. That happens like right away. So we set aside those memory cells pretty much right away when this is going on, so that you know that the next time that you see whatever this is, that your body can protect you against it like way faster, because it has those set aside. And then going back to the T cells, when we activated those guys, some of them can become specialized effector cells. Effector just means like they have special job, right? They do. And those, or they can be memory T cells that kind of, when they get activated again, will be those. Okay. Yeah. So that's the idea. This is how we're going to be looking at all of this. We activate the lymphocytes, and first they will proliferate, and then they will differentiate, turn into do different things. As you see here, like we had from, oh gosh, I just hate, this isn't all in one picture, but essentially we stimulated the B cell here, is a B cell, and then it's going to proliferate and make a whole bunch of them as activated B cells. And then these are what's going to differentiate or change into the B cells down here. That makes sense. So that's what the steps that are involved. Okay, proliferate and then differentiate. How are continuing on with the B cell, not B cells, lymphocyte development in general. When plasma cells make antibodies, essentially here they are just in a certain format called IgM. They get in a like pentamer antibody, and those antibodies go out into the system, the blood, and interact with pathogens out there, and they recognize a specific antigen on the pathogen, a specific part of that pathogen, and you know, stick to it. And what's the point in that? Well, you guys already learned about like complement pathway, right? So you don't need me to tell you about that again. And anyways, then this is humoral immunity. Humoral, remember humor. I just always think of that as the liquid part, but like it's probably not quite the best. But essentially that's dealing with your antibody stuff, your B cell produced stuff. The stuff that's going to be floating around, sort of like a solvent in there. These over here on the right are cell mediated immunity, your T cells, because they're going to actually be cells going out to do work. Okay. That's why they're cell mediated. All right, how do we get different lymphocytes? How do we do all that? So T cells, we know they're going to mature in the thymus. We've said it. They're going to circulate between lymph and the general circulatory system pretty much constant's. They're going to migrate to T cell areas of the lymph nodes and the spleen as well. And then they have T cell receptors as what's called their surface receptors are called T cell receptors. Okay? B cells are going to mature in the bone marrow. There's a whole bunch of them, hundreds of millions of distinct B cells develop as a result of gene modification and selection, which we'll talk about soon. They'll circulate through the blood and locate at specific sites in lymph nodes, spleen, and other tissues, and kind of hang out there and wait for activation. They have immune globulins as surface receptors. It's like antibodies stuck to their surface. And this is just depicting kind of the comparison between the B cells and the T cells. Because that's what that was. I'm sorry if you didn't realize. This is comparing T cells and B cells and kind of how they develop and where they're going to be going in the body, which you should be aware of. Okay, this is a little table for that. Okay, this is specific events in B cell maturation specifically, as well as T cell maturation specifically, and what's going on in what area, and whether they're going to, you know, require whatever trigger and whatever. So this is a good little table to refer back to if you need to kind of see, you know, a little bit more detail in the differences. But like the only real things that I am interested in are maybe this one down here, whether it requires antigen presented with MHC. Okay, that's major histocompatibility complex. We'll talk about what it is soon. The product of the antigenic stimulation. So once it sees the antigen, what happens? And then kind of general functions. Of course, you want to know the general functions of these things, but yeah. And then site of maturation. I don't care about the numbers. I don't care about. No, I don't care about that. Okay. Cluster of differentiation. Cluster of differentiation markers are markers on the surface of these cells that we would say CD whatever. Okay, we just refer to it as CD, cluster of differentiation. That helps kind of tell the difference between these lymphocytes that we're dealing with. And this is more specifically dealing with the T cell maturation. Okay. We have markers on the T cells that have to deal with CD whatever. CD3 surround T cell receptor and assist in binding for T cells. CD4 or CD8 depending on the cell. CD4 is involved in T helper cells, and CD8 is involved in cytotoxic T cells. So you are going to have to know what these are. I know you're like, well, whatever. But yeah, no, you're going to have to know the differences of those. Okay, so for B cells, their receptors, they're immune globulins on the surface. Those immune globulin receptors are composed of 2 heavy chains and 2 light chains. This is a depiction down here on the bottom, but essentially 2 heavy chains, like, oh my God. Okay, and then 2 light chains. This is light. Okay, that's the idea of how they're set up. They're shaped like a Y. That's the immune globulin. 1 light chain will be bound to 1 heavy chain, and then they're kind of attached in the middle by a disulfide bond for a Y shape. They belong to the same families as the T cell receptors do, which the T cell receptors, we had the CDs on the T cells, and now we have the T cell receptors themselves on the T cells. Yeah, they're pretty closely related. They're not shaped the same necessarily, but they have a similar idea, similar concept here. Here are the B cell surface receptors versus T cell receptors. So you see here on the left, we have the B cell receptor over here, and we have MHC2. On the right for the T cell, we have the T cell receptor in green. And then CD3 is going to help with binding to that. And then that's just a thing it does. And then CD4 or CD8 for depending on if you're a T helper or a cytotoxic T cell. What does cytotoxic sound like to you? Cell killer, right? So depending on what you're doing, you'll have CD4 or CD8 on there. So those are the complexes associated with that. There's 3 important ones on the T's. There's 2 important ones on the B's. The immune globulin and which is also known as the B cell receptor. Okay, so we're going to watch this. This is one of my favorites.
People are full of antibodies, a lot of antibodies. These proteins are a vital part of the immune system, but just how the body makes such a variety of them mystified scientists for decades. The human immune system has to fight off an endless variety of bacteria, viruses, and parasites. And for each invader, it needs a specific antibody. Luckily, it can generate billions of them, each slightly different, ready for any disease. But these antibodies, made from 2 light chains and 2 heavy chains, are proteins. And billions of different proteins should need billions of genes to make them. Humans have around 20,000 genes. The solution to this problem was a Nobel Prize-winning discovery. Antibodies are made by B cells. Like every other cell, B cells start off with the same 20,000 genes. But they also have an enzyme called RAG. RAG is a DNA shuffler. It targets the genes that make the antibody's binding site. There are 3 kinds of gene segments here, called V, D, and J. For the heavy chain of the antibody, humans usually have around 130 different V segments, 27 D segments, and 6 J segments. The RAG enzyme binds to one of the J segments at random, and then to one of the D segments, chopping off the spare DNA between them. Other enzymes then join the ends together. The same thing happens with a random V segment. So you end up with a gene composed of 1 V, 1 D, and 1 J joined together. This provides over 20,000 possible VDJ combinations for the heavy chain. The light chain can have another 400 possible gene combinations. Combining heavy and light chains gives approximately 8 million different possible antibodies, each with a different binding site. And that's not all. During the joining, extra DNA is added or removed at the junctions of the segments, which takes our antibody diversity from the millions into the billions. This is how our immune system can respond to diseases it's never even seen before. In fact, there's so much diversity that one of our antibodies could potentially bind to almost any target in the whole universe.
Pretty cool. I think that's some of the coolest stuff I have ever learned. I don't know. I think it's kind of inspiring when you think about it. anyways, let's move on. So this is going to lead us to that concept of clonal selection. Essentially, you have your stem cell, and then you have your kind of precursors for what you're leading up to with whatever cell we're talking about, whether it's your B cell or whatever it is. So you have your stem cell here, and we're going to become, I don't know, B cell, whatever, and it has all these different receptors on the surface out here, right? These ones originally are going to get targeted to see if they can interact with certain self molecules. If they do interact inappropriately with self molecules, we will eliminate those cells. Okay, so right, so let me sure. Right, so we'll eliminate those so we don't have any like anti-self action going on. Like, so that's what they're calling here immune tolerance, so that you're not attacking yourself. That's the idea. Okay, next, what we have are these specificities for these single antigens. Okay, this binds with one thing, the green associated thing, the purple associated thing, the brown, and whatever. They each bind with one specific thing. And then we can have all these guys waiting out and, hanging out in the bloodstreams and whatever. But essentially, like if we had entry of an antigen, whether it's being presented by an actual presenting cell or, typically it's going to be a presenting cell bringing it in there, that's going to trigger these guys to multiply to immune response against whatever that antigen is. So that's how we're going to select down from all of these different choices. So when we say that we had like billions of different possible antigens that we can react to in the universe, I mean, we have billions of these. We have billions of these. So it's pretty interesting stuff. But yeah, so that's the idea of clonal selection. We have a whole big slew of different things we can react to. We pare it down to the ones that seem like they're going to be useful. The ones that aren't anti-self, right? We don't want to keep those. The ones that seem like they are behaving weakly, we can also get rid of those. Yeah. So that we have a whole repertoire, a whole pool. It uses the word repertoire. I guess that's fine. A whole repertoire of lymphocyte clones just waiting to be activated. By a specific antigen. So let's break down what an antigen is. I keep talking about antigens without really getting into detail on what I mean by that. An antigen is any substance that provokes an immune response in a specific lymphocyte. That could be self as well, but anything that provokes an immune response or could potentially provoke an immune response. To be perceived as an antigen, also known as an immunogen, a substance must meet certain requirements in foreignness. So we don't really necessarily want self-reacting ones. So foreignness, shape, how it's going to bind with that binding site, size. Sometimes if it's too small, it won't interact with those little protein molecules. And accessibility. So if it's an antigen that's within, deep within a protein, it can't ever see that antigen and we're not going to react to it. So antigenicity is a property of whatever that thing is, its ability to behave as an antigen. That's its antigenicity. My words today. Okay, so we need antigens to be perceived as foreign in order to react appropriately to them. Unfortunately, sometimes we will interact with self and see it as foreign, or we will see, donor tissue as being foreign when it's not. So that's a problem as well. But anyways, the molecules that is going to be made up of, we get complex forms that should be unique essentially for each of these antigens that we've been encountering, and we'll remove the self-reactive ones during the clonal selection. So what kind of antigens can we react to? They're not just proteins. Of course, we would think of proteins interacting with other proteins, but they can be polypeptides. That's like a protein, like parts of proteins interacting with one another. Little bits interacting with one another. I'm not making full proteins though. Lipoproteins, fat with protein, glycoprotein, sugar with protein, nucleoprotein, nucleic bases, nucleic acids, and bases interacting with proteins, polysaccharides, that's sugar chains, and then lipopolysaccharides, fats and sugars linked together. So let's watch this. I guess, oh, I had to click on that.
Antigens are macromolecules, usually of molecular weight greater than 10,000, such as proteins and polysaccharides. They are recognized by the immune system as foreign. Individual antibodies are not made against the entire antigen molecule, but rather to particular chemical groups on the molecules known as antigenic determinants or epitopes. Many different antibodies can be made against a single antigen, each antibody reacting with a different epitope. Complex structures, such as the surfaces of bacterial cells, may have many different epitopes. Each different antibody binds only to the correct epitope.
All right, so haptens are things that are too small for us to interact, for our antibodies and stuff to interact with, so they're just too small. It can't interact with it and hold on to it and create a signaling pathway as a result. So we can stick those things to carrier molecules in order to get our bodies to react to it and create that stronger sort of reaction. So that's something we can do with haptens. That includes a lot of toxins. In the case of like tetanus toxin, we have to have it stuck to a whole bunch of other toxins. We got it's usually Tdap, right? It's tetanus, diphtheria, and pertussis toxins all kind of stuck together to help each other be recognized by your body. So alloantigens, these are proteins and other molecules of one person that are an antigen to another. So we recognize it as foreign. These are surface markers that occur in some members of the same species, but not in others. So that just means like from person to person, not necessarily from species between different species. The basis for individuals' blood type is included within this. That's alloantigens, as well as there's a lot of molecules that can be, but also histocompatibility profiles. It's very important when you're getting transplants of organs. The HLA profile is extremely important. You want to match it as closely as you can to avoid graft rejection. That's what I used to do for Integris for a while. I used to do testing for that. Super antigens, these are nonspecific. They're going to lead to nonspecific activation of T cells. It's like overstimulation of our T cells. So this any antigen that can do that. They're potent stimuli for all the T cells, activate them at a rate 100 times greater. This includes some bacterial toxins, and that can lead to like toxic shock syndrome. So we have antigen that's going to be associating with our target lymphocytes, right? Antigens will be further acted upon and formally presented to antigen-presenting cells. Essentially by the antigen-presenting cells are going to be like the dendritic cells. I'm trying to monocyte type cells. That's what I was trying to get at. And then B cells can be antigen-presenting cells as well. Essentially, the cell is going to ingest the antigen, migrate to the nearest lymphoid organ, and process and present the antigen to the T lymphocytes to activate them. So, class 1 MHC genes are markers that appear on all nucleated cells. So MHC is that major histocompatibility complex that our body uses to recognize whether something is self. So class 1 MHC genes, these are markers on all nucleated cells. And if you have a nucleus, you've got an class 1 MHC. So that's what we're saying is like if you get a liver from somebody and the MHC from that person is different than your MHC, your body will recognize their MHC as foreign. So that's what the previous information was talking about. But anyways, this is allowing for self-recognition and regulation of immune reactions. Each person has kind of their own combination of class 1 MHC kind of genes that they can combine. And then endogenous, these are going to always present endogenous antigens. So stuff that came from within the cell, like viral molecules that come from within the cell. That's going to be presented on MHC1. MHC2, these are for immune regulation on macrophages, dendritic cells, and B cells, again, the antigen presenting cells. These are going to be involved in presenting to T cells during cooperative immune reactions. That's meaning that the T cells are going to help activate like B cells or whatever to go fight in, the fray or whatever, or make antibody, I guess, and whatever. So they're going to present the exogenous antigen. So if it came from outside of the cell, it'll go associated with MHC class 2. Outside the cell, MHC class 2. Inside the cell stuff, MHC class 1. I'm not too worried about the class 3, but that has to do with complement. Just know class 3 does with complement, okay? All right, so this is just depicting class 1 and class 2 MHC molecules. There you go. So let's get into the T cell response. The best, so when we are undergoing lymphocyte activation, the best cell that will recognize the epitope, it's not really what I wanted to say. The cell that recognizes the epitope best, that's what I'm trying to say, will undergo proliferation and then differentiation. We've mentioned it earlier. They're going to increase in number. We're going to clone ourselves. They're all going to be exactly the same. And then those cells that were exactly the same are going to differentiate. They're going to change into different types of cells. Those cells that have proliferated and then differentiated, they're going to be differentiating into memory cells for remembering next time, effector cells going to be doing whatever their job is, and then regulatory cells. They're going to help with suppressing whatever the reaction is that's going on out there, so we're not getting out of hand. This is breaking down the T cell types. So we have T helper cell type 1s that deal with, they have CD4 with MHC2. They'll interact with MHC2 for stuff from the outside. So essentially that's how that would be interacting. And then other T helper cells, we have CD4 on. So it's CD4. This is just too complicated to be going through this. Let's just go through the slides first. and then you guys are welcome to come back to this chart. It's hard to explain the chart without explaining everything all at once. So let's move on to the slides for them. So T helper cells. T helper cells are exactly what they sound like. They're going to be helping out other immune cells. They are CD4 positive. So that's all you need to know about that CD4 part. T helper cells are CD4 positive. They are going to play a role in regulating immune and reactions to antigens. They're going to be activating and controlling responses by B cells and other T cells. And they're involved in activating macrophages as well, so that they can, kick them into high gear. They usually do this by direct contact. Anyways, they can differentiate into specific kinds, which I was getting into with that table just a second ago. TH1 helpers, TH2 helpers, or TH17 helpers. So those are the kinds that they can differentiate into, and this is going to, I think, better explain what that means, what they are going to do. So TH helper cells, TH helper 1s, we'll start here. They're essentially going to stimulate macrophages. TH helper 2, they're essentially going to be doing B cell stuff. This is a memory one. We can differentiate into that. We're always going to make memory, right? TH17 is going to trigger inflammation responses, and Treg is going to be dampening the immune response where it needs to, right, to help us from going out of control. So all that's going to happen when we have the antigen presenting cell and the CD4 T cell interacting with one another and triggering, activation that way. So that's how T cell activation works. with CD8 T cell activation, so these are effector cells. So those are helper cells. We just went through the helpers, THs. That's helpers only. These are CD8 T cells that are effector cells. What does that mean? They have their own job. They actually do a thing. They're not helping anybody else do a thing. They're doing their own thing. So essentially, we'll have a helper cell come along, and we have our antigen presenting, like dendritic cell or whatever. And then our CD8 cell here will be activated. So now it's activated. When it's activated, it will have differentiated into a memory cell, of course. And then it will go out and interact with infected cells. So this is a cell that's infected, not a pathogen. So if I have viruses growing in my cell, like influenza viruses or something, they grow inside of my cell. This is what this is going to interact with, anything growing inside. Okay, so they're going to interact with stuff presented on MHC1, because that stuff, remember, is from the endogenous presentation. So that's from within the cell. So they interact with MHC1. They have, anyways, it gets activated. It sees it as infected. It sees this as foreignly infected. And then it's going to secrete these two things. These are important to know: perforins and granzymes. Perforins poke holes, they perforate. Granzymes, zyme, breaking down, like destroying the cell. You can even induce apoptosis of the cell, because the cell's going to be like, wow, I'm really getting screwed here. And then it will undergo apoptosis, just killing itself. I just realized YouTube's probably not going to like that I said that, but you get it. Effector cells of the cell-mediated immune, right. So cytotoxic T cell. So that's a CD8 T cell, is also known as a cytotoxic or TC T cell, right? So that's like anything that's going to be going out and killing these other cells. They are effector cells. They go out and kill. They recognize a foreign peptide complex with MHC1 from within the cell and mount an attack on the target cell that is presenting it. This will be things like virally infected cells, cancer cells that are not growing properly, and then cells from other animals or other humans. Killer T cells, like I said earlier, they will make perforins that will punch holes in the target cell and allow for the granzymes to enter. The granzymes will come in and start breaking everything down, and the target cell will undergo death by apoptosis as it is being degraded. So let's watch this video. Or not.
When a virus infects a cell and synthesizes vital proteins, some of these proteins are degraded to peptide fragments. Similar to what occurs with all normal proteins made in the host cell, these peptide fragments are complexed with class 1 MHCs and displayed on the surface of the infected cell. Activated cytotoxic T cells interact with the virus-infected cells by recognizing both the viral antigen and the class 1 MHC. The cytotoxic T cell then releases granzymes and perforins, which cause perforations in the cell membrane and induce apoptosis in the infected cell. Cytotoxic T cells remain intact, detach from the infected cell, and move on to other target cells.
All of these videos, by the way, are in your textbook. So you can access them that way if you would like. You don't have to sit through them on here. Geez, I click on the right thing. Okay, moving on. Okay, so we also have other cells that can help out with all of this, like natural killer cells. They're kind of related to the T cells. Yeah, I don't know. They're not specific for anything. The only ones that are specific for stuff is going to be the T cells and the B cells. So I don't know. There you go. T regulatory cells, they are also considered part of the helper class. They carry that CD4 marker that the helper ones do. They control inflammatory process to prevent autoimmunity. Like if we're getting too out of control reacting to these bacteria here, we might start reacting to nearby cell proteins and stuff like that. So we want to control all that, keep it reeled in, keep it focused on what you're supposed to be focused on. These kind of help out with that. Okay. Get their own little intermediate slides. Right. So I had said earlier about the cell that best recognizes, that's probably the better way, the epitope will undergo the proliferation and the differentiation. This slide, we've already seen it. We applied it to our T cells. Now we're going to be applying it to the B cells. This is an outline of how all of that's going to be working. Essentially, you're going to have your antigen binding at the surface. You're going to process it and present it. And then we're going to have a T helper cell cooperation so that we can start increasing activation and interacting that way so that we can start the whole process, sending signals to kick everything into gear, essentially. Then the B cell is activated. Then we're going to differentiate. And then we're going to go through clonal expansion as a result of, while we're making all those plasma cells and whatever. So what's a plasma cell and what's all that? What's all this mean? We're going to get into the details of this in a little bit. Because I really don't want to get into much details looking at this, but once we do get into the details, come back here to this slide and you can look through these steps here so you can understand a little bit better what's going on, because this is actually like the pared down kind of outlining version of what you would need, what we're going to do. So it's good for going back to like review and understanding.
Antigens, such as bacteria or viruses, are usually phagocytosed and degraded into small fragments by professional antigen-presenting cells. Antigen-presenting cells, also known as APCs, include macrophages or dendritic cells. These small fragments are then complexed with a special type of protein called a class II major histocompatibility complex protein, or MHC, and transferred to the surface of the antigen-presenting cell. Specific helper T cells recognize an antigen when it is presented on a class II MHC because helper T cells have special recognition proteins on their surfaces called T cell receptors. This interaction activates these helper T cells. The activated helper T cells are now capable of stimulating B cells that also recognize the same antigen. These activated B cells become antibody-producing plasma cells.
Okay. So that's a nice little breakdown of it, a simplified version of it. So we're going to get into some of the little details for this. Okay. The FAB. I don't really need you guys to worry too much about the specifics of the makeup of these antibodies. So remember we had talked about immune globulins earlier and how they had the B cell receptors and how they had the heavy chain and the light chain and all like the rearrangement and everything for the different. So those essentially become antibodies. So they will become a thing that gets secreted by the cell, by the B cells. The B cells that do that are called plasma cells. So this is just showing a depiction of what an antibody looks like. And the FAB region is that part that has like the portion that can be all the different things, all the different types of 8 billion different reaction things or whatever. Yeah, so that's the part that's variable. So that's the FAB. This part is the part that's going to determine essentially what type of antibody you're making. So there's a few different types of antibodies out there, and we'll go over them here in a second. But first, another video. No, yes.
Human cells do not have enough DNA to have separate genes for each antibody molecule. Instead, different segments of DNA can be mixed and matched to form different antibodies. Light chains are made from V and J segments in the variable region and a constant region segment, whereas heavy chains are made from V, D, and J segments in the variable region and one of 5 different constant region segments. As the lymphocyte divides during maturation, its genes are rearranged. DNA sequences are assembled by the random selection of V and J gene segments for the light chains and V, D, and J segments for the heavy chains. The constant region of the molecule is encoded by a single C or constant gene segment for light chains and 5 different constant regions for heavy chains. The rearranged DNA in the mature B cell is transcribed into messenger RNA and translated into the light chains of the antibody. DNA sequences for the variable regions of each heavy chain are assembled by the random selection of a V gene segment, a D gene segment, and a J gene segment. The genes for the constant regions of heavy H chains have Greek letters designating IgG, gamma, IgM, mu, IgA, alpha, IgE, epsilon, and IgD, delta. The DNA in the mature B cell is transcribed and translated into the heavy chain protein. The light chains are then attached to the heavy chains to form the functional antibody. Because of the large number of ways in which the genes can be rearranged, many different antibodies can be produced.
So essentially, like we talked about the rearrangement of that one before in that other video that I like better. But essentially what I feel like, oh no. One of the important messages of this video is this part here is letting you know that this constant region codes for or equates to what type of antibody. AB means antibody. Okay, so interacting with the antibody and what it does for us, like what is even the point of an antibody? If I were to ask you guys to write me out, what's the point of an antibody? What does it do for us? This is the answer here on this slide. So I would definitely take home this message that's on this slide right here. Essentially, here's what they can do. Antibodies can coat the surface of a bacterium and prevent it from doing its stuff because it's coated. Antibodies can stick on the outside and or like when they're doing the coating, help out the phagocytes to eat them. That's called opsonization, remember? So you can do that. So that's one thing. This is the second thing. We'll go over here, third thing. Another thing it can do, we can essentially cover like viruses and toxins and other things and enzymes and everything like that. It doesn't have to just be large bacteria or cells. We can also interfere with the function of whatever that molecule is, that antigen is. That can be useful. That's called neutralization. You can read that, yeah, kind of. And another thing we can do is cause everything. Here you can see how that we're using antibody to cause the things to stick together. That's called agglutination. That's number 4. They obviously can't do their thing if they're stuck all together. Another thing we can do is activate complement. That is number 5. We had talked about that as the classical complement pathway when we learned about that in innate immunity. That's what's going to happen here. Poking holes in it and getting rid of it. We can also use antibody as an antitoxin. That is literally like special kinds of antibodies that are specific for exotoxins that are being secreted so that exotoxins themselves can do their thing. So that's like what we're looking for with the tetanus. That's 6. So remember all 6 of those. All right, the different classes of immune globulin, they have their purpose. There's a reason why I was telling you guys about that constant region and the mu and the alpha and all of that. The first ones that are always going to be produced are going to be IgM. We always start out with that first response. It also can be a B cell receptor on the top surface of the B cell. You see how it forms a pentamer? That's an important characteristic of IgM. The monomers are going to be everybody else. The only one that's going to form anything different will be the dimer that we see for IgA. IgA is important because it's for mucous membranes. That's an important one to remember. IgA, mucous membranes. IgG is probably our big old long-term helper immune one. This is like our plasma cells just secreting it out into our blood. Once we have our memory cells and we've got a full-blown reaction to a bad guy, we will stop making IgM and focus pretty much on IgG because it's a little bit better at providing protection. IgE is associated with allergies and worm infections. And then on the surface of the B cells, those typical receptors that we were talking about, that is IgD. So you do need to know what that part of it. We can use immune therapies to target specific aspects of, other molecules in our bodies or other targets that we might want to attack to quiet them down. Let's say somebody has cancer, we can attack cancer cells because they have a specific marker associated with that type of whatever type of cancer it is that you're looking at. They might have a specific marker that you can attack. And I want to say trigger your immune system, but we can make antibodies that can attack that molecule and infuse you with them so that your body can use those antibodies to attack your cancer. If that makes sense. Immune disorders, we can use antibodies to target inflammatory cytokines. So those messenger molecules like the tumor necrosis factor. Tumor necrosis factor, we can have antibodies just coat it so it can't do its thing and cause inflammation. Of course, that's where we would talk about it causing immune suppression, because you need TNF alpha in your immune responses if you are sick, right? You don't want it to be too suppressed. So it's kind of a, balancing game. And then, we can have humanized antibodies that are from humans, or monoclonal antibodies that are specific, one single antibody that only binds with one single antigen. And then we have trained leukocytes, and then, we can use them to help with making vaccines as well. Anyways, so this is Keytruda. It's used for treating all these different kinds of cancers. It will, you get this infusion with this antibody, and your body uses that antibody to, bind to the cancer, and then your cells will react appropriately to being covered with those things being covered with antibody. So that's the idea of how that works. If you guys ever see any commercials for anything that has MAB, adalimumab, for example, which is Humira, it's the next one. That's monoclonal antibodies. So Humira's adalimumab, that is a monoclonal antibody that will bind with the TNF alpha receptor, so that TNF alpha cannot signal. And then that's also why it causes immune suppression. So you got to be careful with all of that. But these are the diseases that it has been approved for treating. I think it's also been approved for a couple other things since this. So a titer, if we're going to get an antibody titer of somebody, we're trying to look at their blood levels of that specific antibody that we're interested in looking at. So you have a lot of antibody in your system right now for COVID, or if you don't, or if you have a lot of antibody in your system right now for, chickenpox. And if you do, then you would be protected from it. Or if you don't, then you might need a booster shot too. So we can use that as a determination of how your immune system is holding on to memory. The primary response to an antigen upon the first exposure to antigen, we have a latent period, and that is because we need to start making antibody. Like we have to turn all of our cells on and like find the right cell to activate and do all the T cells and having the T cells activating the B cells and everything like that. All that stuff has to happen before we can actually start responding. So our first primary response to an antigen is always the slowest one. There's that latent period at the beginning. Because we don't have any memory built up. We don't have enough cells that can respond to that specific antigen, right? So we have to trigger all of that first. So then the plasma cells produce the antibodies. The serum titer will increase to a plateau, of course, and then we don't need to make any more. We're not being signaled to make more anymore, and then we'll taper off to a low level for weeks and months. IgM at this point is switching over to IgG, which is a stronger responding antibody. We didn't have it in the beginning though. The secondary response, which we will call also the amnesiatic response. We see it again weeks or months or years later. Our antibodies start getting made at a lot faster rate than they did the first time, because all we have to do is activate the memory cells to start making those antibodies, essentially. So that's the idea there. We'll make mostly IgG there, as well as IgA in the mucosa, depending on, whatever area this might be going on and all of that. So that's going to be super protective for us and way faster response. And you can even respond so quickly that you don't even know that you were exposed to a pathogen. So this is a layout of how all of that works. This is your latent period over here on the left. We have our primary response, and this is like the antibody, measure. Oh my gosh, I hate it. Over time, so it's just the total one is the dark one. Anyways, so there's that, and then years go by, whatever, we see it again, and then it's just like bam, and now we've got IgG, and then we can very quickly clear that infection a little more quickly than last time, but definitely responding much faster. So yeah, second time you're really not going to see much IgM at all. So that's what that's saying. But I apparently drew on this at some other time and saved it like this. Whoops, I don't know, or somebody did anyways. But if you monitor antibody production over time, essentially you will see that once the same virus will attack the body, like within a certain time frame, you'll see a huge response in production of antibody. So I don't know. This is talking about different kinds of immunity. You can have active versus passive. You can have natural versus artificial. The example in the upper left, we're talking about active. That's your body making antibodies against a thing you got sick from. Passive so that was natural immunity. Natural immunity can also be passive, which would be you passing your antibody on to your baby through breastfeeding. Artificial immunity would be active if we are introducing a stimulus to you that was not introduced naturally. So like a vaccine, and it's not something you encounter naturally. And then now you're protected from the thing. And then passive artificial immunity, that would include receiving immune therapy like IgM or IgG from, someone else that cleared an infection. And now you're receiving that, or receiving immune globulins to treat your cancer against that cancer, like we were talking about in that other slide. So yeah, being delivered from something else. Okay, so we know that our surveillance sort of system for our immune system will be our resident macrophages in whatever tissue you have. Interstitial fluid is where a lot of this is going to be moving around. It's in between tissue, in between organs and stuff like that. We have lymphatic return back up to the heart via, your muscles pushing it. And then we have the lymph nodes where we have things kind of hanging out. So that's where we will have a lot of surveillance going on, in these spaces where stuff did happen. And now we're just going to hang out and wait. A lot of it's going to be in the lymph nodes. Invasion happens again. So we will have tissue damage. We have inflammation because of the cytokines and all of that, and we'll start killing cells nearby. Yeah, a lot of messengers are going to be activated, and then we're going to start responding with our specific response. That includes proliferation and then differentiation. We have our T helper cell activation, so we can start activating more B and T cells. Memory and effector T helper cells. Then we have the B cells that will provide antibody production via the plasma cells. And then we will have T cell, cytotoxic T cell, I always call them CD8 T cells, but cytotoxic T cells that will go out and kill cells that are infected. That's going to lead to tissue repair in whatever areas you're having the infection. That can lead to scarring because your body doesn't care what you look like. It's just trying to contain itself and create another barrier. And then we have memory response that will be developed as a result of this as well, and be maintained in your lymphoid organs in the bone marrow. Vaccines do all of this stuff at the same stuff. They work the exact same way. We have different kinds of vaccines that you can have. Live attenuated is literally what it sounds like. It is alive. It is an actual living virus that has been altered in a way so that it cannot propagate well and create disease in you. It may be able to grow, but it can't create supposedly symptoms. Some people have experienced mutations in these, and it has led to disease. Unfortunately, that happened with polio, but that's really uncommon. All right, so we have killed viruses like the flu. That's dead. It's not alive at all. We have subunit viruses like we have for HPV. That's just parts of vaccines, excuse me. That's just parts of the viruses that you need to respond to. You don't need the whole virus. We have toxoids, which we had talked about previously with tetanus, diphtheria, and pertussis. And we have conjugated, which is putting a couple of things together in order to elicit a response. So how can we get that stuff into your body to get you to respond to it? We can put it in subcutaneously through a shot. We can put it in into your muscle, which isn't just subcutaneous. Subcutaneous is just under the skin, whereas intramuscular is like into your actual muscle. You can also have intradermal, which is like only in the skin on the outside. It's like these little patches that they have, and they often do for allergies. And then we have oral and nasal administration of like the aerosols and stuff. And what are some common side effects? We often have reactions at the site of the injection. Fever, allergies are pretty common, usually to the stuff that's in the vaccine and not to the whatever you're giving the vaccine for, not to flu, but to like the egg that it was used to make it. Because your body is eliciting a response against flu. And so it's eliciting a response against sometimes egg as well, because it's nearby in the area, unfortunately, and then you become allergic to eggs. That is possible, or maybe you're already allergic to eggs. I don't know. And then you respond to it inappropriately, but these are possible side effects. So we introduce our antigens by whatever means we just said. Our body will process it and present it. We will have our T helper, cytotoxic T cells and B cells respond, proliferate, and then differentiate into memory, which is what we're really going for with vaccines. And then we have all these antibodies produced. Normally that would clear an infection. We don't need to clear an infection in this case. We're just trying to trigger development of memory. Infections sometimes can elicit a stronger response than a vaccine, and sometimes vaccines can elicit a stronger response than infection. It just depends on what we're talking about. This is a thing called herd immunity. Herd immunity essentially says, like, we have this thing called R0, okay, and it has to do with how long the virus or whatever the pathogen is infectious, the disease is infectious within somebody, how much contact that people might have that would be sick from this, and the probability that someone who is infected with this would infect others. So, you know, if the lower the R0, then there's ranges on this. So it depends on what R0 we're looking at out here, but depending on that range for that thing, we might not be so concerned that it would be spread very easily. However, like if you look at like whooping cough and measles, those have a pretty high R naught. That means that they're going to be highly contagious and spread very easily. In the case of that, you would need to have a higher incidence of vaccination in order to prevent other people from getting sick in your community. So you can determine how many people in your community might need to be vaccinated in order to provide actual true herd immunity to protect them, based on this and other data as well. And this is just an example that they give. Measles, you need to have 93 to 95% of the population vaccinated in order to prevent the others who can't get vaccinated from getting ill from that disease and preventing spread from it. And eventually, hopefully, lead to, elimination. But these are some common misconceptions associated with getting vaccines, that you can get too many too soon, that you can have vaccine injuries. Some of these things are, a few of these things can be somewhat accurate, but they've been blown to proportions that are not helpful to anybody. One of the concerns, which I think we had talked about previously, with it containing baby parts, that was a big issue with the COVID vaccines. I remember that just has to deal with the fact that we used HEK293T cells, which are human embryonic kidney cells, to study and grow and develop the virus and et cetera for making the vaccine. And the things used to make the vaccine. Those cells have been used for a very long time, decades, in a lot of different things that you've already enjoyed thoroughly, I assure you. Does that, that opens a whole other can of worms that I don't want to get into right now, but that's a misconception. It's not accurate to say it contains baby parts and that babies were killed for these vaccines. That's inaccurate. Not for those vaccines. Natural immunity is always better. No, I just told you guys that it's not always. If you can survive whatever the illness is, I mean, good luck on some of these. If we're talking about smallpox and 80% of the people would die today, good luck. Do they consider smallpox vaccines, for those of you who are out there saying, oh, I'm a boomer and I'm proud of it, and I got my smallpox vaccine because they stopped giving them a while back. I don't have one. Or maybe you're in the military. Smallpox vaccines are only considered effective for 10 years. So, sorry. Good on you if you have it. I think it is a good idea for smallpox vaccines. I'm just saying, personal opinion. Bioterrorism and all that. But you definitely want your soldiers to get those vaccines for sure, because they're going to be in the front lines of it, but if it were ever used. But moving on, I'm getting off the rails here. Toxins, they all contain these toxins. Some of them did back in a while back, but not really the ones that anybody's ever talking about. They don't have. So it's sort of weird that they're getting it mixed up that way, but I don't know. It's whatever. And oh yeah, and your daily, like day-to-day life has way more mercury in it than those vaccines did. I promise you, you're not avoiding it in your day-to-day life. And if that disease is not around anymore, or some people argue that maybe it never existed and it was just, big pharma trying to pay, whatever. Those are common misconceptions. I'm not going to get into the anti-vax movement with you guys. But we know that this is all over the place, and I know that you guys are aware of it, and that is really all I really want to say about it. And if you want to talk to me about it, I'd be happy to talk about it with you. It just not right here. Right, same old deal. I just can't. Okay. All right, common side effects. So we're talking about common side effects of this stuff. Depending on that, you do want a little bit of response, right? If your body is responding, like if I told you just now about all the inflammation and everything that goes on when your body is responding correctly to an antigen in the immune system and everything, and that you need to have all this inflammation occur. If you got a vaccine and you didn't have any side effects, I mean, it might be working just fine. It's possible. But if you have side effects, that's just telling you that it is working, FYI. So you responded to it. There was a discussion a while back about the measles, mumps, rubella vaccine causing autism. If you haven't heard of this, I don't know what rock you were living under. The actual study that the person published has been retracted, and it is because it was falsified information. It was falsified results, and nobody has ever been able to replicate what he stated in that paper. Just FYI, everything about it was false and it was a lie, and it's caused I don't even know. Let's move on. Let's move on. Anyways, you know what it's caused, guys. All right, so what are we talking about? The third line of defense, it's the specificity, right? We want to delay, it's a delayed reaction, unfortunately. So that's why we like that we have our innate immune response to kind of cover things until we can get this going. It's a strong response though, once we get that specific response going, and we can develop memory, which is important. You don't get that from the innate immunity stuff. So, you know, B cells differentiate into plasma cells that produce antibody, and T cells, they are associated with cell-mediated immunity. They can be helpers or they can be cytotoxic, right? There are 5 types of antibodies. You should know what they are and where they are, what they're for. And then vaccines are the same immune system as every other disease. They're not any different. So it's not, and you shouldn't be reacting differently than that. So that's that. And we're going to be done with this. And I will see you guys for chapter 16.